It's my privilege to introduce Ms. Donna Muzny, Assistant Professor at Department of Molecular Human Genetics at Baylor College of Medicine, also Director of Operations of Human Genome Sequencing Center, and Division Director of the HEAC Clinical Laboratory. I just want to say that I was really excited to have Donna here today with us, complementing very nicely our first talk. We're going to move more into kind of clinical. So some of the questions we had were like, so in the clinical space, what can we do with this, these new assemblies or reference genome assemblies? That's why it was really important to get someone like Donna and her expertise at this meeting today. So just as a brief background here, Donna is a primary member of the BCM HESC faculty, so Baylor College of Medicine Human Genome Sequencing Center, and has been there since it was founded in 1996. And at that time, it was selected by the National Human Genome Research Institute and chose it for one of the 6 pilot programs for the final phase of the Human Genome Project. So you're kind of getting this really nice perspective of when this whole thing started to now up to 2022. Currently, as the director of the Human Genome Sequencing Center Clinical Lab, and just want to emphasize clinical, her interests have expanded into clinical assay development, management, and management to provide large-scale production delivery of targeted next-generation sequencing panels, exome, genome, and RNA-seq applications for clinical research within a CAP and CLIA environment. So again, I'm not going to do all that justice, but the point is that there's tons of things that go into bringing, again, a lot of what we heard this morning into now clinical settings, and we're excited to hear more from Donna during her talk. And again, I could sit here and list her accomplishments, but I think I'd probably take a couple hours to do that. Just are really, really too numerous to list. Donna's published over 400 publications, of which have been cited over 100,000 times. So it's a really huge honor to have her here today, get her perspective on this really important and timely topic. And then just one little thing I wanted to highlight is Donna won it. So Best Female Scientist Award this year, 2022. And again, just as a testament to her, the impact she has had on this field. So without further ado, I'd like to introduce Donna to tell us all about genomic data flow for all of us.
Thank you for that wonderful introduction. Yes, I'm going to speak today about the All of Us program and the impact that may have in the coming years upon return of results in a clinical environment, but also the data that we're generating is going to be there for all investigators to mine. So let me just go ahead and start. I'm also going to talk first a little bit about the Human Genome Sequencing Center, give a little perspective about the Human Genome Project, and then go into the topics around the All of Us program. So the Human Genome Sequencing Center was established in 1996. We have about 180 staff, and the primary projects that we've done, of course, is the Human Genome Project, but also the Human Microbiome Project. We've also participated in TCGA, Cancer Genome Atlas, and what we try to do is to take emerging technologies and optimize those platforms for high throughput production, and then take them and put them in the clinical lab once they're hardened. And this is the cycle that we've been through for many years. Right now we have 12 NovaSeqs, we have 5 PacBio sequels, 2 Prometheans, and so you can see that we have a lot of capacity to take on these larger projects. So in the early days, in the beginnings of the Human Genome Project, it started about 1989, but it established 2 principles that are still in place today. One was the open participation of a group of scientists from all different countries, and and working collaboratively. So it was a large-scale genome program, a large project. The 5 major institutions that generated the high amount of DNA for this program, of course, was Baylor College of Medicine, Washington University in St. Louis, Whitehead at Boston, the DOE centers at Walnut Creek, and also, of course, the Sanger Center. The second really important thing that it did is to establish the Bermuda Principles. And what that was is the sequence that is generated, all investigators agreed from all of the countries submitting and centers submitting that the data would be released to a database within 24 hours. So those 2 things were really guiding principles of the program. And of course, from 1990 to 2003, we generated the data, and but it not only was for the 3 billion bases that made up the human DNA, but also we identified the 20,000 or so genes, identified variants, and also completed a number of model organisms along the way. This is an early meeting in Cold Spring Harbor. You see Francis Collins, an early Richard Gibbs, George Weinstock, Eric Lander, and Bob Waterston. I'm sitting right there, and this is Jane Rogers. So very historic, and also in 2003 we finished it. There was a public effort and there was a private effort, and I would have to say that the activity by Craig Venter at Solera kind of propelled us to finish ahead of time and below and under budget. So since the completion of the human genome, we at the HGSC have been completing genomes. We first started with James Watson, Desmond Tutu, Jim Lebsky, and it's really moved from doing individual variation to more and more looking at medically actionable type of disease-causing variants with Jim Lebsky, investigating his Charcot-Marie-Tooth syndrome, and the Berry twins, where we found medical management and implementation of to modify the progression of their disease. And now we've been focusing in the last many years of doing tens of thousands of both genomes and exomes to understand genetic architecture. And these here are just a few of the projects that we have tackled, moving from large data sets of common disease to more rare diseases here with the Greger and Mendelian programs, and of course the UDN, Undiagnosed Disease Network. Really key to this expansion in what we're sequencing and tackling projects has been the decrease in cost from the 2.7 billion that we spent to do the first human genome to now really a genome is less than $1,000. And if you really just want to capture the coding regions of a genome, an exome you can get by with about $250. So our next movement forward, the next step, of course, is going to be the sequencing of a million participants in the All of Us Genome program. Some of the things that I'm going to talk about is the generation and logistics, the genome reporting. There's a clinical or a report. It's a research report, but it is a genetic report that you will get if you so participate in that portion. We had to work under FDA compliance, and now, like I mentioned, that we're opening up the data for general scientists to use for their research. And also now, we're going to start into generating thousands of samples with long-read data from the same data set. Key here has been recruitment, and right now we have over 500,000 participants, 300,028 with electronic health records, and we have about 400,000 biosamples received. Here, this is the recruitment strategy, and if you look, trajectory here, and if you look we were on a great trajectory, and then the pandemic hit right here through this period where we leveled off a bit, and right now the recruitment is starting to pick up again. And in fact, there's going to be 2 surge recruitments, participations in the next, one next week, and I believe one at the beginning of December, to get this number of samples really going up again. So the All of Us research program, of course, is to gather health data from 1 million or more people throughout the United States. The Genomics Workflow, with what we're going to do, is to return ancestry from the genotyping arrays that we're generating, and reporting back with ACMG 59. These are incidental findings in your clinical that are medically actionable genes, as well as 7 genes known for drug interactions from the whole genome sequencing. Both of these are from the whole genome sequencing. So the milestones to date has been establishing a joint workflow between all of the centers that are involved, and obtaining an FDA investigational device exemption. This was key to starting the program, and I'll talk a little bit more about that. And behind that, because to accelerate recruitment, we also came back and did an FDA IDE supplement for saliva samples. Key also has been a data freeze for 98,000 whole genomes. Part of this has been written up in an analytical validity paper of how to approach FDA and validate such a large device for FDA approval, and now the researcher workbench. So to date, we've completed 330K genomes and just over 550K genotyping arrays. So what is this AOURP, all of us research project, Genomics Network? What does that look like? How does it operate? Well, first, the participants are recruited. They give consent. They're sampled for blood and potentially saliva. There's other collection processes involved as well. Mayo Clinic is our biobank, and Vanderbilt is our data resource center. So what happens is the samples are de-identified at Vanderbilt and then proceed back through the biobank to the different genome centers, which includes Broad, which partnered with Color for reporting. The HGSC, we partnered with CIDR at Johns Hopkins for doing the GDA arrays, and University of Washington. So both array and whole genome is generated. We have a clinical and interpretation process, and then reporting back to the participants if they consent. Digging in a little bit deeper, what does this process look like? Well, for whole genome sequencing, these centers generate the data. For whole genome sequencing, the same centers, some of us generate the genotyping arrays. These genotyping arrays are not in the clinical area, and they, if you want, you can get back your ancestry and some specific traits from the genotyping arrays. From whole genome sequencing, we built a return of results pipeline where you can receive your PGX report and or your medically actionable disease panel. Now, I won't go through this, but this is what we had to get through in order to send samples between the centers and the information between the DRC, Mayo, and each of the genome centers, and then to the groups that are doing the interpretation. So it's complicated, but this is what took a great effort to get right between the centers. Just a little bit on our production. We have, thus far, the HGSC completed 100,000 of the 330,000 whole genomes. And I put some milestones on here with the SALAVA approval, data freeze, researchers workbench. And most recently, we have completed 100,000 samples at the HGSC. That happened just last Friday. We had a little celebration on that. Some of the thresholds that we look for are common across the centers to QC the whole genome data. We spec'd this out and validated it at least 30x coverage. And these are some of the other metrics that are really key. One, of course, is contamination below 1% as compared to other human data. And then mapped reads, especially for SALAVA, we benchmark no lower than 75% of the reads must map back to human. So the process for returning the genomic results is also involved, and over the summer, quite a bit of work has been done to navigate that between the centers. After the data is generated, the clinical validation takes place, and so this is the identification of likely pathogenic variants, and then validation of those with an orthogonal validation device, such as we're using Sanger, other groups are using another panel, NGS panel, to validate. And then color is then doing a report harmonization platform, and whereby the reports are generated, and again, color does the genetic counseling. But this report and harmonization platform is really key. It's important that the centers across the different groups have the same variant classifications, and they're regularly updated. And then it also allows for a common sign-out of environment. And then this engagement module back to the participants is really important so that they get their data. Right now, we're in the middle of doing our first releases of the reports. This started back in August. We had friends and family, and we're now in the 950 and ramping into the 1,000 participants per week, where we're going to review the reports and then issue both the PGX and the HDR, or hereditary disease report, on the ACMG 59. It starts with an informing consent loop to make sure that the participants still are able to, interested in receiving these data. And thus far, we've processed over 200 reports for both the HDR and the PDX. So one of the items that was really different is this FDA IDE validation, and it's really key for us to be able to get launched. Because this was a direct-to-consumer sort of process, that means it's not under CAP, well, it is kind of, you have to do your CAP-CLIA, but you don't have an ordering physician. Anybody can sign up for it, and it's one of the major reasons that the FDA decided there was significant enough risk that we needed to validate and do an IDE through them. Over 1,000 patient and control samples across the 3 centers were sequenced and used to validate and show equivalency on both the data, the quality of the data, the variant calling, and the reporting for all of the centers. The device in this instance means that the device starts at the beginning to the end. That means from the initial consent through the delivery of the data, the report. We needed to prove through accuracy, precision, limit of detection, invalid rates for the sequencing, and if any interfering substances from the collection devices could impact the validation or the results. So in these cases, the FDA and what they require is way above what we do for any normal CAP-CLIA lab. And it takes quite a bit of work for our groups to continue. Any updates has to go back through the FDA for a supplement, a 5-day notice, and of course an annual report. And we summarized this information in a paper that was published last year. And so if you're interested in any of that, we laid it out pretty much what it takes to to get through an FDA application. This is one of those diagrams kind of charting out how many times we interacted with the FDA and how it took basically 19 months to execute the validation and get it through the FDA. And we had many interactions with him. Here at the end, you can see that we had, it's summarized, we had 52 requests, and a lot of them, the FDA comes back with questions when you finally do your submission, and most of those had 24 hours that we'd have to turn around. So now, every time we do a supplement, we pretty much have to go through the same process. We have to be on call for whenever the FDA has questions so we can get it through. Future types of genomic data reporting for the AOU program. We're going to extend the gene list to the ACMG 78. These listed genes just came out as being medically actionable. We would like to add the reportable range for PGX to include CYP2D6. Other variant types. Right now we're not reporting on CNVs. We want to expand that. And also different types of genes, including a potential polygenic risk score. Like I said, all of these would have to go through an FDA formal change management process. Now going into a little bit about what we've done to release the data that we've generated thus far. We have a tiered access hub, and the 3 tiers are public tier, where there's just general data stats available on aggregate data, and this is available just by going to the all of us websites. And then there's a registered tier, gets into some of the electronic health and wearables, and different surveys and so forth. And then there's a controlled access tier. And this is the data set where you have to be approved. You have to be approved here, but this is a more rigorous approval to access genomic data and the genotyping arrays. So a little bit closer look at what this data looks like. 98,000 whole genomes, 165 genotyping arrays were released in March 2022. And the variants are called, and you can browse through them as well, 600 million, just about. And then there's different combinations of the data available to go through with different phenotypic information that has been collected to execute any research. Now, the research has to be done on the researcher's workbench, but different types of tools are going to be supported all in a cloud-based format. Now, one of the things that this genomic data set is, it's really racially diverse, more so than a lot of other data sets, databases out there. Here is the self-reported ethnicity, having over 20% African American and Hispanic sets. When it's computed, it's pretty much very close to what the self-reporting is. So this is really is a rich resource. One of the things that we have done so far is take a look at the data through different various demo projects. This one has been done by Eric Venner in our group. And here he's looking at predicted pathogenic variant frequency by different racial genetic ancestry. So here he's found that both the African, Latino, and European they're different by a chi-square test. And so more work is going to be done to look at this, but this is just an initial look. So finally, one of the things that we're really excited about, and I'm glad Sergey gave an overview of all the long-read data and the different platforms earlier. One of the things that we're really, really excited about, and we've spent quite a bit of time developing as well, and to put into production mode these long-read platforms. We have now targeted really 1,000 samples for ONT and 300 samples for PacBio. These samples are just 1 year. This actually doubles if we're going to plan out for the project. But basically, the sample selection targeted Hispanic samples from this 98K WGS release. So all of these already have Illumina data. Fritz in our group used his XV collector tool to maximize our allele representation in this data set. And basically, we are going a bit deeper than some of the other groups because we want to get the best representation of the SVs, structural variants. 25x genomes here for ONT on one flow cell. And with PacBio, we're also going 25x, and this will be 3 SMRT cells. Also a target length that we're now achieving of over 17 kb. And this just details some of the different kits that we're using. Both of them have been released most recently, and that took some work to get those into production this summer. We now have 5 sequels and 2 Prometheans, so all of that capacity, most of that capacity will be directed toward the long reads for this program. So in closing, some of the genomics deliverables that I talked about was the genotyping, the whole genome, participant in the return of results when you're consented for return of results either for PGX or this hereditary disease region, the researcher workbench, and we are anticipating our next data release, whole genome data release in early 2023. We're going to be looking at over 300K genomes to be available. And then for the program, across all of the genome centers, we expect over 11K, 8x PacBio, about 830, 25x PacBio, and over close to 4,000 ONT WGS. So with that, I'd like to thank, of course, our AOU, my AOU research program colleagues, basically organized into 3 main groups for technical, regulatory, and interpretation and reporting. So these are all of the colleagues at the different centers. And then, of course, I'd like to acknowledge Richard and Eric Burwinkle, who have been tremendous leaders for our Genome Center. So with that, if there's any time, I'll take some questions.
Hi, that was a great talk on kind of like how it all fits together. And one of the things that's really interesting about the long read technology, it's kind of frustrating but also is exciting, is like how do you balance like they're constantly changing, especially in nanopores. Like every week is like, oh, we just updated the base caller, it's like 1% more accurate. How do you balance that with like locking in a standard operating procedure for production?
You're absolutely right. That is an issue. And right now we're fortunate that it hasn't moved for the last 3 or 4 months. But yeah, right when we decided, or this opened up this opportunity to sequence all these genomes for AOU opened up, everything moved on both platforms. So it is that is a really important point. It is also really important in that if you're going to go clinical, this thing has to stop moving because you cannot afford to validate over and over and over again. Not for a CAP CAP-CLIA clinical lab, and definitely not for the FDA. You'd just go insane. So you're going to have to pick your thread of what are the most important, and when an important release comes up that you might want to change for, that will enable something like CNV calling and stuff like that for some of the softwares.
Any other questions? Donna, I have a question. That was a wonderful talk. So let's say right now I use Virco, for example, and I have a new assembly that's the best that exists. How long would it take to get that new assembly incorporated through these processes? If I would say, oh, I have a new genome assembly, it sounds like there's a lot of validation and everything that would just, is there a time frame estimate that
So if you're talking about software like to do the mapping and so forth.
Oh, or just say there's a new human genome reference.
Oh, new human genome reference.
That's I say I have, and it's great, and I hand it over right now. How long would it take to get it all through the?
If you're doing research, it won't be too much time. If you're doing clinical, it will take a lot of work, because the annotations for clinical interpretation have to follow. Right now, we're still doing interpretations on Build 37. And all of the data that I talked about is on Build 38, and so we're on a dual pipeline for AOU almost, just because of there's other things that have to follow.
All right. One last, we have probably time for one more question if anyone has. If not, let's go ahead and thank our speaker for a wonderful talk again. And